So what are the changes that are happening in cells? Not chemical compositions, but the physical content as the cells progress from a benign stage to aggressive cancer phenotype. Now, this research that I'm talking today falls within the third research thrust that I have in the lab. The first one is the development of new micro/nano fabrication technologies. The second thrust is to use these new fabrication techniques, and we develop microanalytical chemistry instruments, and including microgas chromatography. And one of the biomedical applications of that we're pursuing, that I'm not going to talk about it, that's a completely different research topic, is to do point-of-care devices, to develop point-of-care devices for breath-based diagnostics. And if you don't know, the breath has a lot of compounds, around 2,000 compounds, that can be indicators of different diseases, including breast cancer, lung cancer, and diabetes. But that's a different subject. And the third thrust is, again, to use those technologies that we develop along with the other technologies that have been developed in the micro/nano engineering area. And we go ahead and we analyze cells, including cancer cells, and we see what is the physics behind cancer progression, and whether we can use those to do cell manipulation, including bacteria. But again, that's a different subject I'm not talking today. But today, basically, my focus is primarily on cancer biophysical characteristics. And when cancer happens, you have a positive feedback in your system that's gone wrong. So there are biochemical events that are happening. There are changes in the cell cytoskeleton architecture and the overall cell structure, including cell sizes and cell membrane properties. They can induce changes in the cell biophysical characteristics, and at the same time, you can see alterations in the function of the cells, including the growth pattern, the viability, gene expression, so on and so forth, and then it can cause cancer progression and metastasis. And when such a positive loop basically is created, then if you use drug, you basically create a negative feedback in it, and you try to close that loop and then make it into a more stable situation. Now, and that, again, that drug treatment can also affect the properties of cells. Now, in the metastatic process, what happens is that the cells escape from the initial tumor. They go through several mechanical deformation as they go through pores, and they get to the blood vessels, and then they escape from the blood vessels, and they form a secondary tumor. All of these cases, cells goes through changes in its biophysical microenvironment that can affect also the cell properties, as I show you later. But cells at their healthiest states, they have a very organized cytoskeleton architecture, but then they go into a disease stage. Specifically, the actin filaments become disorganized, the esterase fibers loosen up, and that can affect the properties at the single cell level when it comes to biophysical characteristics of these cells. The two cancer types that we have been focusing in the lab has been ovarian cancer and breast cancer. So for ovarian cancer, which is diagnosed very late actually, and the mortality rate of this disease is pretty high, we have used a model, a progression model that has been developed by two of our collaborators at Virginia Tech, Dr. Roberts and Dr. Schmelz, husband and wife. And they developed this mouse model that can be, that has these cells at different stages of this disease, from very healthy state to very aggressive phenotype, and also they have been able to extract tumor-initiating cell-like cells or cancer stem cells out of these aggressive phenotypes, and we have also characterized those properties as well. But these cells almost match the human cell lines phenotype characteristics, and a very good model that we have used in this case. The second type has been breast cancer. Again, around 12% of women develop invasive breast cancer cell, as you see here. And there are a lot of mortality cases, again, caused by breast cancer. And for this, we have used the human breast cancer cell lines in these experiments that I show you. And in particular, we have used 184A and MCF10A as indicators of normal cells, and then we have used 468 and 231 as moderately invasive and very highly invasive breast cancer cell models. The two techniques that we're using are primarily atomic force microscopy and microfluidics. So atomic force microscopy and microfluidics, we can go ahead and at single cell level, we can go ahead and characterize the properties of the cells. And these properties include the biomechanical and bioelectrical properties of these cells. And then we want to see whether these biophysical markers can be used as a reliable source for cancer detection, diagnosis, monitoring the drug response, and even the relapse of cancer if it happens. Now, talking about atomic force microscopy, if you're not familiar with that technique, in atomic force microscopy, you have a cantilever beam, and this cantilever beam, you can go ahead and send it in close to the cell. And then you can go ahead, what you can do afterwards is that you can go ahead and deflect the cell. It's again a single cell. You can deflect it, you can deform it, and then by measurement of the amount of deformation, you can determine what is the Young's modulus or mechanical properties, or other way, a spring constant of the cells. And what we do is that we attach a micro bead to the tip of this cantilever beam so that we can apply almost a homogeneous force on the nucleus of the cells. And basically what you do is something like this. Yeah? So you do, and then you see whether the cell likes it or not. So how much resistance you get from the cell as part of this deformation that you apply. And then you can generate graphs, and then you can use models to extract the properties. So you can have the approach phase, then you have indentation when you go ahead and poke the cell, and then you can leave the cantilever there, and then you can let the cells to start relaxing itself around, and then applying a force so that the bead goes up, and then you can retract it. So from this you can get the viscoelastic and elastic properties of the cells. Now, the results that we got, this is the mass ovarian surface epithelial cells that I described are most cells. And if you look at the graph, basically shows the early, intermediate, and late stages of cancer. And if you look at these graphs, which shows also statistically significant results, the cells, as they go from a benign stage to a more aggressive cancer phenotype, they start to become more deformable. They lose their resistance against deformation. They become more deformable. And as you see, the elastic modulus goes down. And also the apparent viscosity also goes down. They become less viscous, which is associated with the transient behavior of the cell as a response to the mechanical deformation. So what it means is that cells, as they go from a benign stage to a very aggressive phenotype or cancer phenotype, they become more and more deformable. So part of this metastatic process that I told you, cells have to deform themselves and go through pores. Normal cells, they're stiffer. It's very difficult for them to go ahead and squeeze themselves, to deform themselves. For cancer cells, they have lost that ability, and because they become deformable, and they can go and diffuse through pores and go. And then there was another study that we did, and we looked at the different cytoskeleton organization components, and we realized that the actin filament organization has really significant effect on the cell mechanical properties. And if you look at these graphs, for example, on the top you see the most earliest stage, and you can see the actin filament very organized, and then you can see the most late, and you don't see that organized cytoskeleton architecture as you see it in the earliest stage. And again, that contributes to the biomechanical characteristics of these cells. We did the same study in breast cell lines, and we saw the same thing. So as you go from a normal to a very aggressive cancer type, again, the cells become more deformable, and they become less viscous. So the same kind of behavior we observe in the structures. The other thing you can see is that the cancer cell lines, the distribution in terms of the mechanical properties is more uniform compared to the normal stage, which means that mechanically, or the biophysical characteristics of these cells, when they become cancerous, it becomes more homogeneous compared to the normal status. And as I told you, the actin filament really plays a significant role in reducing the elastic, or basically reducing the deformability of these cells. When then treated the cancer cells and normal cells with drugs, with sphingolipid metabolites, and treatment with sphingosine, which is an anti-cancer agent, we realized that improves the actin organization, and at the same time, as you see in these graphs, it starts to increase the spring constant of these cells, makes the cells stiffer. So it has a positive effect on cancer cells, makes them less deformable when you go ahead and treat them with this anti-cancer agent. On the earliest stage, you don't see that much effect of this sphingosine on the biomechanical properties of the most cells. Now, if you go ahead and use tumor promoting drugs, the ceramide, or if you go ahead and use sphingosine 1-phosphate, you see the opposite effect. You see that the cells becomes more deformable. So these two chemicals that they are almost abundant when the patient has ovarian cancer, they cause the cells to become stiffer. So this chemical microenvironment can also trigger the changes in the mechanical properties. And these are, again, showing all the studies, and these are all published, so you can go ahead and take a look at the papers if you want and see. Again, this shows the same effect that as you go ahead and treat them, if they become stiffer and stiffer, they become more heterogeneous in terms of their biomechanical characteristics. As I told you, we also did tumor-initiating cells. We studied those, and this was basically the first publication that appeared that basically our group basically initiated that one, that we started to look at the biomechanical characteristics of cancer stem cells at single cell level. And as you can see, the cancer stem cells are subpopulation of very aggressive phenotype. And so you see the late, and then you can see cancer stem cells. And if you look at those, then again you see that the cancer stem cells are even softer than very aggressive phenotypes. And if you go ahead and treat them with sphingosine, they make them stiffer, but not to the level which makes them biomechanically close to normal stage. So which means that, again, the cancer stem cells are very, very deformable. And even if you treat them, biomechanically still they are categorized in the aggressive stage. You don't make them stiff enough to make it difficult for them to migrate and to metastasize. So that doesn't happen in these cells. And our collaborators have really put about 100 of these cancer stem cells into mouse. And the tumor has grown pretty rapidly, and the mouse has died very quickly. So these are really, they have the characteristics of cancer stem cells. And again, as you see basically in the graphs, I don't know if I have a laser pointer, but this one you can see that these are much softer than the late stage, and much softer than the early stage. Now, this is a very recent study that we have done, unpublished. I presented it last week at the Nano Engineering Conference for Biomedicine and Chemistry, and that one, again, I thought that might be good also to present it here. So the other thing that we did, which was pretty interesting, is that instead of poking the cell one time, that a lot of researchers, they do, what we did here is that we applied, we kept it there, then we retract, and then a minute later, we applied the second force. And then we did this repetitive or cyclic mechanical deformation. And this is more natural compared to just doing one time and then you go away. Because again, as I said, through the metastatic process, you see that they go through this continuous deformation and relaxation phase. And we got something very interesting in fact. So these graphs, they show the behavior of cancer cells. These are showing the behavior of normal cells. So if I want to say it in simple words, the cancer cells, as you go and deform them more, they become a little bit softer. So they like it. They don't resist against it. Normal cells, if you deform them the second time, they become more resistant. So it's almost a natural process that they resist against further deformation, which means that if they want to escape, even if they go through the first deformation into the microenvironment that they're living in, the second time, they need to spend more energy to deform themselves. And at some point, they don't have that energy, and then they just don't advance. Yes? But cancer cells, they basically like it. So again, which means that even if you treat the cancer cells, what is more important probably is to bring them back these deformation characteristics. So basically, you need to give them more resistance. So the fact that they're soft is bad, but the fact that they become softer or they don't increase their resistance is very, very bad. And again, that's again another study that we're doing to see whether we can go ahead and predict what happens to the cells when we go ahead and add the drugs to these cells, and whether the biomechanical characteristics are changing when we do the drug treatment following several or cyclic deformation. And this also has single cell value. So if you look at the normal and cancer cells, on average, normal cells are stiffer. But if you go ahead and take a look at the single cell values, there is a significant overlap between the behavior of normal cells and cancers. About 80% of the cells, they display the same behavior. So even though it has important implication when it comes to the average population behavior, you cannot basically go ahead and extract the biomechanical properties of the cell, and just based on that, you say the cell is cancerous or normal. You cannot do it. On average, you have 1,000 cells, you can say whether it's cancer population or normal. But you cannot go ahead and detect the single cells. When you go ahead and do multiple deformation and you look at the slope of changes in the biomechanical characteristics, that has a significant value. And you see here that when it comes to normal cells, both the elastic properties and the viscoelastic properties, they display positive slopes. When it comes to the cancer cells, at least one of these properties, they show a negative slope. So you can almost define a cancer region and a non-cancer region. And here, using this algorithm, we have been able to identify about 90% of the cells as being cancerous or non-cancerous. And in fact, that the number has been higher, about 95% is the confidence level that we have. So it has a very interesting single cell value when we go ahead and do this multiple deformation. And a few slides that I have on a new technology that in the lab we have started to investigate out of an NSF grant that we got last year. And this is based on microfluidic technology. Now in this case, we have the inlet and outlet. That's where we introduce the cells. Then we apply negative pressure to this port, and we are able to get single cells coming through this constriction to this pore, and then the cells gets deformed, and then we are able to characterize the transit time as the cells pass through. Not going through the fabrication. So this one, you see this cell that's going through, and this is another one. See how it is for this cell to go through, and how difficult it is for this one. This is a cancer cell. This is a normal cell. The cancer cells, it's easy for them. They deform themselves, they go travel, and they go to the other side. For the normal cells, it's much more difficult to go through. Which is, again, similar to, again, as I told you, it's a more natural environment compared to atomic force microscopy. In atomic force microscopy, you're dealing with adherent cells. Here, you're dealing with flowing cells. So very, very two different areas of studies that we do. And then later on, we put electrodes. So these are electrodes, and we're able to measure the bioimpedance of the cells from here to here. And this is the graph, the electronic signal that we're able to generate. And it's very interesting because this electronic signal can determine the entry time and the travel time through the channel. So by just looking at the electronic signal, we don't need to do microscopy imaging. We can just use, this is almost a label-free microfluidic system. That we can just look at the electronic signal and we can go ahead and extract the overall transit time of the cells through this. The other thing about the impedance is that we can go ahead and determine the electrical properties of the cells. And the electrical properties of the cells are related to the cell membrane properties and to the cell cytoplasm properties. Okay, and we are able to extract those properties by just looking at this signal. And this is what we got. So these are the entry time for these normal and cancer cells. These are again normal, these are cancerous. Yes. And these are the cell size and the axial ratio we have basically characterized them using video microscopy. These ones, the two top graphs are extracted using the electronic signals that and we have validated that with the video microscopy. And you can see that, for example, the normal cell here can transit pretty fast. Interestingly enough, the 468, which is moderately invasive also, can transit very slowly, So the same thing that you have for normal, you see that the same thing for 468. And that's almost the same thing that you can see with the matrigel or the migration assays that biologists use in the lab. When they use 468, they also see that a lot of disparity and it takes a long time for them to go through. And that's exactly what we observed here. But the 231s, it's pretty rapid for them. And these are the impedance characteristics of these cells, which is again pretty interesting. If you look at the green one, which is 184A, and you look at the blue one, which is the red one, which is 231, very distinct electrical properties at single cell level. Very distinct. So the red and the green, there's almost no overlap. You look at the blue and the rest, and the blue is moderately invasive. You go to a different frequency, and remember, this impedance, so we can get multi-frequency information out of it. Very rich information, in fact. If you look at that one, the blue at high frequency has a different characteristics, which makes it very interesting because at single cell, again, we can go ahead and separate it from the red, which is 184A, and the red, which is the very aggressive cancer, breast cancer cells. So it has single cell. The only distinction, the only one that we couldn't distinguish between these four is MCF10A. For that, there's a solution. We know how to do it, and we've started doing it, but I'm not presenting it here. So hopefully, the next conference, we can show those results. But we are able to really separate all these four cell lines with single cell value information. So what I'm showing here is that the biophysical characteristics of these cells has rich information, whether it is biomechanical or bioelectrical. And we can utilize MEMS, microsystems, microfluidic, lab-on-a-chip technologies, and nanotechnology, including atomic force microscopy, to decipher these biophysical characteristics and then use them as markers. And as a possible marker, just to give you an idea, is that you can go ahead and do in vitro drug efficacy analysis. You can go ahead and you can extract cells from the patient. You can go and do drug screening outside and see whether they're changing back the biophysical properties into normal stage. And if you're not able to do it, it may not be a good drug to apply to the patient after all. So we can look at these biophysical markers as a possible method to screen for the efficacy of the drugs. And that's one of the current pending NIH proposals that I just told you the idea. So, and these are and I have a lot of, obviously, collaborators both in the microanalytical chemistry and in the bio, nano, MEMS and microfluidics area. And there are other researchers that we do, including the bacteria and pathogen trapping. But again, I didn't have time to include those in these slides. But that's all, and I would be happy to answer any questions that you have. Thank you.
Please.
You're talking about the cancer type? So let me make sure that I... Mhm. Yes, yeah. Yeah, these are cell lines. But the other thing that yes, so the mouse ovarian surface epithelial, most cells, they're almost representing what you get from PDX, patient xenografts, yes? So that those most cell lines are not basically cell lines, yes? They're almost extracted from mouse directly and then implanted, and then we applied it. So they're more similar to a case that you see from the patients. They're not really cell lines that we extract. So in those cases, basically what we're saying is that the cancer cells, the cells basically, as they become more and more cancerous or they become basically more aggressive, they become more deformable. It's become easier for them to penetrate through tissue, through pores, and get to the blood vessels. So if you want to prevent metastatic process, probably one possible way is that instead of trying to kill all those cells, that has a huge side effect. Yes, one of the issues with the chemotherapy is this huge side effect that you have, is that maybe there is another paradigm shift in the treatment of cancer, is that you may be able to change them biophysically, to just don't give them the ability to escape, make it hard for them, and contain it, at least contain it. So that's the whole hypothesis that we have in this one. So.
Okay, short question.
In physiological life, this is how I remember how the red blood cells, at least in the spleen, go through from the intravascular catheter to the outside. The endothelial cells are in layers, but there's a very tiny space, the space about 0.5 micron. And the red cells that are normal are able to squeeze. And so the question they had experimental before you is, we show 8 microns, typically diameter, for what I just said, as far as tumor cells being able to squeeze through even smaller.
Yes, so what happens over there is that you need to know is that these junctions that you have, yes, they are able to break that junction. So there's another study that we're doing, I didn't bring it here, is that we create 3D hydrogel structures, collagen hydrogel. And what we do is that, again, we go ahead and look at the angiogenesis problem. And then over there we see that how these cells basically they start to penetrate through even the gel and create and then break it and then diffuse. The next study that we're doing actually is that we go ahead and put HUVEX or even endothelial and epithelial cells, and then we look at how cancer cells are starting to break those junctions. So over there, because they're on a very soft environment, that's a different story, because they can break it. And when they break it, basically they can create more gaps between the cells, and they're able to go through. So the initial gap might be pretty small, but they can work on it. They can work on it, and they can find a way to go through.
Can we talk offline? Sorry about that. So, thank you.
Yeah.